Well, hello and welcome to this presentation. In here, I'll show you how you can use Xdrop for characterizing the functional heterogeneity of your cell populations. And first, starting with an outline of the presentation, I will start out by showing you Xdrop and how you can generate single-cell assays using this instrument. And then I'll show you some examples of different functional assays performed with Xdrop. And then I'll finish with a summary. And the different assays that I will show you is a cytokine secretion assay, a cell killing assay, and a granzyme B release assay. So problem with traditional conventional bulk assays is that they really miss the differences between the different cells' functionality within the population of cells examined. And this means that if performing potency assays of immune cells in bulk, for example, it is not really known from the results of such assays how many of the cells were actually killing their targets, how many secreted a specific cytokine or granzyme or other effector molecules, and to which level of activity were they acting the different cells in the population. And this means that with the bulk assays, you really only get an average information about the functionality, whereas you, of course, with the single cell assays will get single cell resolution. This meaning that you can characterize in depth the functional characteristics here of the different cells in the populations, and you're also able to sort out the ones with the desired functionality. And the way that Xdrop makes single cell analysis possible is by using double-emulsion droplets. So these are the droplets depicted over here, where you have your cells inside an oil droplet, you have media on the inside, and media also on the outside, growth media. And you will have CO2 and oxygen able to cross the oil membrane here, whereas larger molecules such as cytokines secreted, for example, are contained within the droplets here and can be analyzed after incubation. So in this type of double emulsion droplets, where you have medium on the inside and on the outside, and CO2 and O2 permeable across the membrane here, allows really for extended incubation times, and the platform here also allows for analysis using standard flow cytometers and sorters or microscopy, for example. And importantly, it's possible to recover live cells after incubation here based on the functionality. So with the Xdrop, you can make 750,000 double emulsion droplets per sample with 8 samples in parallel, and this only takes 8 minutes. So one of the advantages with these double emulsion droplets is the very small picoliter volume you have inside here. This means that factors such as secreted factors like cytokines build up fast inside the droplet here. So you'll have sensitive detection of the secreted factors, and you'll also have fast readout compared to larger well-structured. So the workflow using Xdrop here is that you start with your cells of interest, and then you encapsulate your cells in the droplets during these 8 minutes here. And after encapsulation, you'll incubate your droplets with the cells inside using a standard CO2 incubator, and then you can perform your analysis afterwards, such as flow cytometry, for example. So this is how the Xdrop looks, and this is also how the Xdrop sort that we also make looks. And the difference between the two instruments here is that whereas both of them can generate the droplets, you can also with the sorter actually sort based on a single color being green. This is the cartridge where the droplets are made inside. In this cartridge, you'll simply load your sample with the cells inside in this well here. You'll load your oil in this well here, and then you'll have your outer media solution here. And then during the droplet generation process, you will have your droplet generated and collected in this large well down here. As you can see, you can make these 8 lanes in parallel. And Samplex, of course, provides the cartridges and also other consumables for producing the droplets, such as the oil itself and stabilizing solution for the droplets and so on. So now I'll talk about the first assay here, which is cytokine secretion assay. So this assay builds on a originally bulk assay, where we'll have some capture antibodies that binds to the surface of the cells of interest here. And then we'll also have some detection antibodies, and both of these antibodies are directed against a specific cytokine of interest in the assay. You'll take the cells and the different antibodies, and you'll incubate these cells and reagents doing droplet encapsulation here. So you're encapsulating the droplets, and you'll have single cell encapsulation here, where you have cytokines produced by the cells will be captured by the cells themselves, and then detected by the detection antibody with a fluorophore attached. And the smart thing about that is that you will really have single cell resolution, because there will not be any crosstalk between the cells. So a cytokine produced by one cell will not be bound by capture antibodies, which is linked to other cells. And then after incubation time here, you will break open the droplets. You will wash away the unbound antibody, and then we'll perform flow cytometry to look at the level of cytokine expression for the individual cells. And this is how the results of such an experiment look. So here we have taken T cells from PBMCs and stimulated these cells to produce TNF-alpha. And as you can see over here on the right side, where we have the single cell assay by Samplex, we are able to detect after activation here a population of cells secreting high levels of TNF-alpha, whereas the majority of the cells do not express TNF-alpha after stimulation. So when we compare that to the bulk assay, we can see that in the bulk assay we get an average increase. It looks like there's an average increase in the secretion from the cell population as a whole compared to the non-activated ones, whereas we can see in reality the majority actually do not express the cytokine when activated, whereas only a small fraction of the cell does. So this is a graph showing you that we can perform these assays both in singleplex, looking at one cytokine, and also in multiplex. So here we have looked at NK cells and T cells activated, and looked at both TNF-alpha and interferon gamma. And as you can see here, there's basically no difference in the level of detection, irrespective of whether we do singleplex analysis or multiplex analysis. So in this graph here, you can see we did the experiment 5 times. So the results are very similar. And again, these are results for T cells non-activated and activated, as well as NK cells also non-activated and activated. What you can appreciate here is that when we activate the T cells, you can see with these assays, some of the T cells produce TNF-alpha and some produce interferon gamma. And with this single cell resolution, you can see that actually the cells that are producing the interferon gamma, the T cells that are producing interferon gamma, are also producing TNF-alpha, but not vice versa. And this is the reverse pattern seen when looking at the NK cells. So we can appreciate here with the single cell resolution that for the cells examined here, the cells that are expressing TNF alpha are also expressing interferon gamma, whereas this is not the case the other way around. What you can do after such an assay is that you can actually take your cells of interest, the high secretors here, and you can expand them in culture. And if we do that and we then re-stimulate after 2 weeks of expansion, we can see over here in the results that the highly potent secretors retain their ability to secrete the cytokine of interest here after 2 weeks of incubation. And this is just to show you how the cells will look after recovery from the droplets. So you can see we have these are K562 cells examined here. We have a high level of viability right after droplet breakage. And then we'll have rapid cell growth and expansion with little or no lag phase afterwards. So now I'll move on to show you the cell killing assay. This is an assay where we co-encapsulate effector and target cells and look at their interaction. So in this case, we have taken NK cells and stained them with a green cell stain. And we've also taken their target cells, K562, and stain them with a red stain. And then we incubate them together in the droplets with a death stain, with a death marker here. Propylium iodide have we used here. And then we incubate for some time before we do analysis. And this is how the microscopy images can look like after such an assay and after incubation. So here we'll have bright field image showing 2 cells inside. And we can see over here that one of them is a natural killer cell and the other cell is a cancer cell, and we can see that the cancer cell is dead. So it's important here, of course, to keep the cells separated before they actually meet inside the droplets. And we facilitate that by using a well insert here that keeps the cell separated until they actually meet in the droplets. After incubation, this is how the readout from flow cytometric analysis will look like. So we can appreciate here that we have 4 different populations of droplets. Over here we have a population containing only target cells. Down here we have one containing only effector cells. And up here we have an interesting one containing both effector and target cells. And we also have a population down here just containing empty droplets. When we look at these co-encapsulating droplets here containing both effector and target cells, and we look at the PI intensity in these, we can see, as expected, that IL-15 stimulation of the NK cells increases the killing ability of the cells compared to non-stimulated NK cells. What we can also see is that we, with this assay, is able to differentiate between fast killing effector cells here, medium and slow acting effectors. In the assay, we also have the other populations available containing only the single cells. And those provide as nice controls for the condition of the cells throughout the experiment. And when we look at these individual populations here, we can see that for the NK cells, the cell death doesn't increase during the 24 hours of incubation. And this is the same for the KFA562 cells for the first 4 hours of incubation, and we see a slight increase here to around 7 or 9% after 24 hours of incubation. Now the final assay I will show you is the granzyme B assay. So this is an assay where we have a substrate for granzyme B, which is co-encapsulated into the droplets together with the effector and target cells. And the substrate we have here, on this we have a fluorophore and a quencher. And when granzyme B is released inside the droplets, the substrate is cleaved and the fluorophore is released and can be detected by flow cytometry. So again, here we can gauge on the double-emulsion droplets. We can look at the different populations of droplets here. And the one that's really interesting here is, again, this co-encapsulating population here. And if we look at that, we can see that after 30 minutes of incubation in the IL-15 stimulated cultures here, we can see we have an increased amount of ratio of droplets containing granzyme B to around 27% after just half an hour of incubation. It's a really fast readout assay, and you can also see that this level of the level of granzyme B containing droplets increase even further after 4 hours of incubation in the stimulated population here. And this is how microscopy images look of the granzyme B assay. So we can see here that we have some highly positive granzyme B droplets here. If we take a look at one of these, we can have a look inside and see that they contain an NK cell here, and we have K562 debris out here, and as well as the granzyme B itself. So this assay you can combine with a kill assay, and if we do that and again look at this co-encapsulating population up here, we can appreciate that we can differentiate between different populations of cells within this gate. So we can have populations of droplets that only contain cells but no granzyme B. We can also have a population of cells that contain granzyme B without any dead cells. And then we have a population of droplets that contain granzyme B and dead cells, as well as, of course, also a population of droplets that contain no dead cells and no granzyme B. And if we look at the results of such an experiment with these NK cells examined here, we can see that after stimulation with the IL-15, we have an increase. These are the striped graphs here. We have an increase in droplets containing both granzyme B and a dead cell after 30 minutes, and that this increases further after 4 hours of incubation. You can also see that we have an increase in the proportion of droplets containing granzyme B alone after 30 minutes here, and this increases slightly after 4 hours. And we can see that this increase in granzyme B alone seems to precede the increase in granzyme B and cell death in the populations here. We can also see that in this experiment here, we have no increase in cell death alone without any granzyme B detected here in the droplets. So now I'll round off with a summary. So what I've shown you here is that you can use Xdrop from Samplex to make single cell assays. I've shown you some different examples of single cell functional assays that you can use or that you can produce using Xdrop. I've shown you the cytotoxicity assay as a kind of cell-cell interaction assay, and I've shown you cytokine secretion assay, granzyme B release assay as example of secretion assays, and I've shown you how you can combine the different assays here. I've also shown you how you can analyze using flow cytometers and FACS, and you have seen how cells can be recovered and expanded after analysis. And lastly, I've shown you that it's a very easy process to make a single cell analysis using Xdrop. You have single-use cartridges and the oil, which is provided by Samplex, and you basically just add your cells and your assay reagents and perform the assays. So with that, thank you for your attention. I look forward for the Q&A.